We ended our last lecture with something called a monohybrid cross. Now, this is just a fancy word to say that we are looking at the cross between two individuals with respect to one trait. So we did two different examples. We did the purple flower color and we did the freckle example. And those examples just looked at one particular trait. So a traditional true monohybrid cross is actually a cross between two individuals that are heterozygous for that trait. Now, this is analogous to the cross that Mendel performed with his F1 generation. But generally, if I say monohybrid cross, it means we're just looking at a cross for one trait. So I wanted to do a couple more quick examples. Basically, there are three potential outcomes for a monohybrid cross. We did at least one of them several times in the previous lecture where we did big A, little a cross, little a. So let's do the other potential crosses here. Big A cross, little a, little a. You can probably already see what's going to happen. Remember that these two traits here indicate the potential gametes. So those alleles are going to separate into gametes, and one is going to get big A, the other is going to get big A. Likewise, in the other parent, one is going to get little a, one is going to get little a. It's the only option. So here, all of our individuals are going to be heterozygous, and they're all going to show the dominant phenotype. So let's do our other example. Big A, cross little a are heterozygote individuals. So remember, write out which gametes we produce. They both are going to produce the same complement of gametes. When we write this all out, fill it all in, you might recognize this is our 3-to-1 ratio, our 3 dominant to 1 recessive, and then our genotypic ratio of 1 homozygous to 2 heterozygous to 1 homozygous recessive. Don't forget to write your ratios out, and don't forget to label what your ratios actually mean. So when we look at all of Mendel's information, we can deduce a couple of laws or sort of governing ideas for patterns of inheritance. Now, the first one we've already talked about. I don't know that I've specifically named it, but we've been doing it as we do our Punnett squares. And this is the law of segregation. Now, the law of segregation says that two alleles separate during gamete formation. So for last example, where we were doing, let's say, the big A cross big A, little a, these represent parent 1 and parent 2, and they're each going to produce their own gametes. When this happens, during the process of meiosis, each gamete that's produced gets one copy of the chromosome, and therefore one of the alleles. Keep in mind that this diagram shows only one chromosome. If this was humans, there would be 23 types of chromosomes and 2 sets each, so 46 chromosomes. So this law of segregation happens for all of your chromosomes. All of the alleles on homologous chromosomes are going to separate independently. But what happens if we want to look at two traits together? So let's say we want to look at seed shape, whether they are round or wrinkled, and seed color, whether they are yellow or green. What happens when we start looking at two of these traits? Well, it turns out that we have what is called the law of independent assortment. Now, the law of independent assortment says that when you have two different genes on two different chromosomes, each pair of alleles is going to separate independently when gametes are formed. Now, that is a mouthful. What this basically says is getting one version of a particular gene does not affect what version you get of another gene. So let's say that we have a chromosome that contains gene Y. This is going to be for green and yellow pea color. And let's say we have another chromosome. Now this is non-homologous, so it's going to have a different shape, different genes, different sequence. And let's say that one contains our allele for round or wrinkled. This law of independent assortment says that the Ys are going to segregate into their own cells, in their own gametes. And the Rs are also going to segregate into their own cells. And which Y you get does not affect which R you get. So essentially, we're looking at all of the possible combinations of Ys and Rs that we can get in our gametes. So it turns out there are 4 combinations. I'll show you how that happens in just a minute. Let's look at our Ys first. So let's say that our Ys segregate. Now, one cell is going to get big Y. The other cell is going to get a little y. Now, half of our cells have to get the big Y and half of them have to get the little y. So out of 4 cells, 2 are going to get big Y and 2 are going to get little y. The law of independent assortment says the R's are also going to segregate independently, and we're going to get any combination of R's and Y's because getting one version of Y does not affect which version of R. So let's say we assort our big R, little r. We can assort them there. We can also produce this combination here. So any combination of Y and R together. This is our book example of law of independent assortment, and it looks really confusing, but it's actually not too bad when you break it down and look at what's happening. So they have labeled their chromosomes a little bit differently. I'm sorry about that. But it is a way for you to see what possibilities you can have. So larger chromosome contains the gene for color, and smaller chromosome contains the gene for seed shape. Now, during the process of meiosis, we are first going to separate homologous chromosomes. Okay, so if we look at just the Ys, we have pulled apart our large chromosomes, separated the homologous ones. Same thing with our Rs. So over here, let's look at just one side. We are now going to separate sister chromatids. And when we do that, this cell is going to split, and you'll see that this cell is going to produce a little y, little r combination, and then another little y, little r combination. On the other half, we've got our pink ones. And when we separate those sister chromatids, we get chromosome with a big Y, chromosome with a big R. Same thing, the other one. So here is 2 of our combinations. We can get the little y, little r, or the big Y, big R. But remember, there are 2 more combinations that we saw. Let's say these chromosomes line up a little bit differently. So now when we split our cell in half, when we separate our homologous chromosomes, we get a little y with a big R, and we get a big Y with a little r. So now we already have different combinations. And then when we separate our sister chromatids, we get cells that are little y, big R, and cells that are big Y, big R. And so now we have our other combination. Now remember, the other part of this is that not only do we get all combinations, but getting one version does not affect getting the other. So there is an equal chance of getting all combos. Okay, so 1/4 of the gametes will always be big little y, but little r. 1/4 will always be big Y, big R. 1/4 will always be little y, big R, and 1/4 will be big Y, little r. Keep in mind that these are on non-homologous chromosomes. All right, so let's look at how we actually figure out some inheritance patterns here. How can we predict what is actually going to happen when we cross these individuals? I like that this book shows the color and the shape together. It helps us to see what is going on. So if we look at this cross up here, we've got true breeding parents for 2 different traits. The trait is going to be yellow and round, crossed with green and wrinkled. So the only version that we can get in our gametes is what the parents have. So these are our true breeding parents. These are going to produce our heterozygous offspring. We are now performing what is called a dihybrid cross, or a cross for 2 traits. And the traits we are looking at are color and shape. I'll work this problem out in just a minute to show you how it happens, but if we look at all of these combinations here, you'll see that 3-to-1 are yellow to green, and we see the same ratio with the round and wrinkled. 3 round to 1 wrinkled. But we have all kinds of combos in there, right? So although we have 3-to-1 for each of the traits, if you look, we have different combinations. Here we have a yellow and round. Here we have a yellow and wrinkled. So let's actually work these problems and see how much of each of these traits or combinations we get. So really quickly, I want to work out the top part of that last diagram that they didn't show you. Or at least they didn't draw out. When we do a dihybrid cross, the first thing we need to do is determine our gametes. Then we're going to cross, and then we're going to determine our ratios. Okay, so if this is parent 1, the possible gamete combinations are big Y, big R. That's it. The only combinations of those 2 traits you can get are big Y, big R. Likewise, the only gametes from the other parent you can get are little y, little r. So if we cross these, the only thing we can get are heterozygous individuals. So there's our parent generation, there's our F1 generation. So determining the gametes there is actually pretty easy. Doing the cross is pretty easy because there's only one possible outcome. And then our ratios are easy because we're just going to be all yellow and round. And they're going to be all heterozygote for both traits. The other one, the F1 generation cross, the dihybrid cross for these 2 traits is going to be a little bit more complicated. Now I'm guessing you probably cannot look at that and right off the bat determine all of the gamete combinations. So I'm going to give you a little trick, and this trick is a math trick, you probably remember it, called FOIL. And you do this when you're multiplying parentheses, and we're going to apply the same principle here. So for the parent on the left, we're going to FOIL the gametes. So first, the first Y with the first R. So the first gamete is big Y, big R. Outer combination is big Y, little r. Inner, little y, big R. And then last, little y, little r. Now the other parent is going to be the same. First, big Y, big R, big Y, little r, little y, big R, little y, little r. Now, you might guess that this is going to get a little bit complicated, and you're right. So here, let's draw out this big 4x4 square and just start filling it in. So here we are going to get big Y, big Y, big R, big R. Now I know that the gametes are written Y's and R's mixed up, but when you write them in your Punnett square, please put the Y's together. One, it's easier for me to grade, and two, it's easier for you to determine the phenotypes later. So here we have big Y, big R, little r. Big Y, little y, big R, big R. And then I'm just going to keep going. All right, so through the magic that is the Surface Pro, we have completed our Dihybrid Cross here. Because this is a Punnett square, you have to give me ratios. In this case, I'm not going to ask you for the genotype ratios, although it would probably be good practice to do this once or twice just to work on keeping your stuff straight. But we do need to figure out the phenotype ratios. So I'm going to use different colors. So starting with yellow, going to indicate here, this is going to be yellow and round. So I'm going to just make a little yellow and round for all of my yellow and rounds. Now remember, yellow is dominant. So if we have at least one copy, we're going to be yellow. Round is also dominant. So if we have at least one copy, we're going to be round. Yellow and round, yellow and round. So I would suggest when you do this that you figure out some way to mark or indicate. Now you can do whatever you want. You can make stars, circles, cross it off, whatever, whatever works for you. So there's all of our yellow and round. Let's do our yellow and wrinkled. So I'm going to do yellow and wrinkled as a star. So remember, wrinkled is recessive. So we need 2 of the recessive copies. Now let's do the green and round. So here's a green and round, where green is recessive. And then last but not least, we have green and wrinkled. So if we count these out, we will discover that we have 9 round, yellow and round, 3 yellow and wrinkled, 3 green and round, and 1 green and wrinkled. So just like that 3-to-1 ratio, that was really important. This 9-to-3 to 3-to-1 ratio is also important. When you do a cross where both individuals are heterozygous for both traits, in other words, the cross that we just did, you are always going to get this 9-to-3 to 3-to-1 ratio. If you have a situation where you have different combinations of homozygous and heterozygous, you are not going to get this 9-to-3 to 3-to-1 ratio. But if you see this ratio, then you know that both of the parents were heterozygous for both traits. So going back to our nice, neat diagram from the book, we see that phenotypic ratio of 9 to 3 to 3 to 1. Each trait on its own shows a 3 to 1 ratio, but when we look at the combos of the traits together, we get this 9 to 3 to 3 to 1. Let's do another example of a dihybrid cross. Let's go ahead and do another example of a dihybrid cross. Remember, the first thing that you're going to do is to determine your gametes, and we're going to do this using foil. Then we are going to make our cross, and then we are going to make our phenotype ratios. So let's start with the one on the left. This is our individual who is heterozygous for both traits, for yellow and for round. So first gamete is big Y, big R, outer, big Y, little r, inner, little y, big R, and last, little y, little r. Then we will foil the other ones as well. You might notice that we're going to have 2 possibilities and only 2 possibilities. All right, so let's go ahead and foil these. So I would like you to attempt this, to pause the video and attempt it. I am going to pause the video, fill it all in to save a couple of seconds on the recording, and you can turn it back on whenever you are ready. All right, hopefully you have your chart filled out. You can check it with mine, or you can just sort of follow along. So let's go ahead and fill in our phenotypes. I'm going to use the same notation, yellow dot or green dot. So for the first one, we have yellow and round. This whole first row is going to be yellow and round. Second row, we've got yellow and round, yellow and wrinkled, yellow and round, yellow and wrinkled. Then after that, we switch over to green. So we have green and round, this whole first row, and then green and round, green and wrinkled, green and round, green and wrinkled. So for our phenotypes, we have 1, 2, 3, 4, 5, 6 yellow and round, 2 yellow and wrinkled. Now the order here doesn't really matter too much. And then we have 6 green and round, and 2 green and wrinkled. So of course I asked you to reduce, so it would be 3 to 1 to 3 to 1, and then make sure that you fill in the phenotypes as well. Again, the unreduced form is not wrong, just by convention we reduce it. And then of course you could write this in any order as well. All right, so what happens if you don't know an organism's genotype? Well, we can do something called a test cross that allows us to figure it out. So let's say that you have 2 plants. Let's say you have a plant that's purple, and you want to know if it is homozygous or heterozygous, because you want to cross some plants and get a specific color out of your cross. So you want to know what that purple is. In a test cross, we can cross the unknown plant, in this case the purple one, with a recessive. So in this case, the recessive would be a white plant. Okay, so let's look at that. If we cross an unknown purple, so we don't know what the other one is, with little p, we can have one of 2 outcomes. If that unknown plant is homozygous, all offspring will be purple. let's say that that plant is heterozygous and we cross that with the white one. In this case, in case you haven't memorized this one yet, this is the one where we end up with 50-50. So we have 2 purple to 1 white. Then you actually let your plants fertilize. So you actually perform the cross, the live cross, perform the fertilization. And then you look at the phenotypic ratios. So if you get 50%, if you get 50% purple, you know you had a heterozygote. If you get all purple, then you know that you had the homozygous individual. Now let's look at a second way to do the test cross. So let's say that we allow the plant to self-fertilize. If we have a plant that is homozygous dominant and we allow it to self-fertilize, again, we're going to get all purple. If it's a heterozygote, what ratio do you expect? This is our heterozygote, cross heterozygote. That's going to give our 3-to-1 ratio of 3 purple to 1 white. So then again, we can allow our individuals to actually produce seeds and then work backwards to deduce what we have. If we see 3/4 purple, then we know that that plant was heterozygous. So let's look now at our book's example of the test cross. So if we have an unknown genotype for the round seed and we let it self-fertilize, we can look at the offspring. So if they are both, if our generation is heterozygous, then we would expect the 3 round to 1 wrinkled. Or we can do our test cross with the recessive phenotype, the little r. And again, if the individual is heterozygous, then we're going to get 1 round to 1 wrinkled. So in this case, we predict the expected ratios, and then we look at the actual data to see which set of ratios our data fits. And that's how we can deduce the genotype if we don't already know it.